Okay, so I have the pleasure to introduce the first trainee, who is Soodeh Moallemian. I'm sorry if I mispronounce. And she is a PhD candidate at the GIGA-Cyclotron Research Center in vivo imaging at the University of Liège.
Hello, everyone. My name is Soodeh, and thanks for having me today. Today, I will present my work on multimodal imaging techniques for AD biomarkers. I will start with some good news. Life expectancy during the last 2 decades have increased. Now, imagine that all of us are above 80 years old, even older. There are several studies showing that we will somehow have to deal with Alzheimer's disease, dementia. By 2015, more than 153 million people over the globe either will be AD positive and we will need people to care. So it is important to study this disease. But what are the biomarkers of Alzheimer's disease? Amyloid beta aggregation and tau accumulation in the brain are the main risk factors for Alzheimer's. But other risk factors such as sleep disorders, family history, and education also play their own role. In brain, microstructural changes start 15 to 20 years prior to the first cognitive impairments. In case of AD, I can name a few here. First is demyelination, and then iron accumulation in the brain. Other neurodegenerative factor is synaptic density loss. Studies proved that there is a co-occurrence of these 3 biomarkers in the brain. So today I will focus on these 3. Thanks to PET imaging, Bassan and colleagues showed that using UCBH radio tracer, we can index the density of synapses in the brain. On the other hand, using NPM MRI, we can derive quantitative maps such as MT-map and R2-star, which relate to tissue properties like myelination and iron accumulation. Today, I will answer 2 main questions. First, I want to see if we can characterize patients from healthy controls using different image-derived biomarkers of AD. And then I want to investigate if using all these biomarkers in a multivariate model, we can see the difference between the groups. To do so, we will use a data set of Alzheimer's patients and healthy controls that all underwent a PET and MRI scan. To talk about data processing, all data were brought to the brain structure bits, and with some modifications on qMRI toolbox, the data is processed. Here, for this study, we are only using gray matter maps that are normalized to MNI space. The PET processing is done in SPM12. From the first frames of dynamic PET, we masked the seed for carotid artery and a mask for carotid is created. Then, using a Pearson correlation between the seed and mask for carotid, we calculated the input function for kinetic modeling that is used in PMOD software. In PMOD software, log-on graphical analysis is done on the PET scans that were corrected for partial volume effect. And in the end, we get some VT maps that show the distribution of radioactive compound in the brain. All VT maps are brought to MNI space. As I mentioned, all maps are in the same space, so we are good to do some univariate analysis. But each map and modality here represents a different property and unit. So we cannot use these maps in a multivariate cohort. And they are not comparable like this. What should we do? Z transformation. We did Z transformation on the data over each modality, overall subjects. Now we have the data ready for doing the statistical analysis. We did the univariate GLM in SPM and then then tested for the difference between the two groups. For the univariate general linear model, we controlled for age and sex. We also did multivariate general linear model using MSPM toolbox, which is a recently developed toolbox that works under SPM. And again, we controlled for participant age and sex, and we tested in presence of all modalities having the same weight if we can find a difference between the groups. Here you can see the univariate GLM results for empty saturated maps, showing us a difference between AD and HC in the right hippocampus. But for the iron map, R2 star maps, we couldn't find anything significant, which might be due to small sample size here. For PET VT maps, our results support the previous findings of Bastan and colleagues, showing the difference between AD and HC groups in bilaterally in hippocampus. The multivariate analysis results in bilateral difference in hippocampus that were significant at voxel level. Here in this slide, you can see how each modality contributed in our multivariate model, showing the empty SAT maps having the most contribution, while the R2 star has the least contribution. We cannot say anything about the direction of these contributions yet. To conclude, I can say that we can use quantitative maps to assess AD in its early stages, and alterations in myelin content and synaptic density occurs in hippocampus in early AD. Furthermore, take into account the interactions between myelin, iron, and synaptic density gives us a better understanding of the difference between AD versus HC. In the end, multivariate GLM model is preferable to multiple univariate models to test for AD because it is believed that AD is caused by a cascade of processes, not just one. Thank you for your information, and you can have the abstract here.
Thank you for your talk. That was really interesting. Do we have any questions in the chat?
No questions yet.
Okay, I have one actually. So I was wondering, so I'm not familiar with this type of PET scan that you used, and I wanted to know if in future studies you're planning to also do the same kind of approach, but using maybe a tracer for tau or amyloid?
Yes, our AD patients were diagnosed using amyloid PET, and UCBH radio tracer binds with SV2A proteins that sit on the synapses of neurons. And when neural degeneration occurs, synapses density, we lose the density of synapses, and UCBH measures the SV2A protein on synapses. So here we are using this radio tracer. But you are right, tau protein also can be used.
Thank you.
And there's one more question in the chat. So it's from Mustafa Bourara, who's asking, I am wondering how can those changes be reliably attributed to differences in myelination, given the non-specificity of MT to myelin, and same thing for R2 star, which is not specific necessarily to iron.
Can you repeat the question, please?
Yeah, so how can the changes you observed in AD with MT and R2 star be reliably representing myelin or iron?
Well, MT-weighted images are proportional to myelin content in the brain, and iron is and R2 star maps are also proportional to iron content in the brain. And I don't get the question yet. How can I say that these are related to AD? Is it the question?
And also, I think he was mostly asking about the lack of necessarily specificity. So it's not just myelin, for example, that impacts MT.
Oh, yes, sure. There are several risk factors that are there for AD, but we cannot investigate all risk factors in one model because it is hard to have all these factors. Yeah, sure. If it was me, I think it is better to have the sleep information of the participants too, or other factors. This is the data that we got.
Okay, thank you. I think we're going to move on to the next trainee.